I'm Fiona Blazsovsky and I'm a life science expert at Atos and a biomedical engineer. Today I want to talk here from my home office about a very relevant topic in our current situation, the fight against Covid-19. There are a variety of different approaches, but today I want to talk about 3 main points.
The outbreak of the novel coronavirus represents a pandemic threat that has been declared a public health emergency of international concern. In this webinar, I would like to highlight 3 main steps in the fight against this pathogen and the relevance of the combination of disciplines like bioinformatics with technologies such as high-performance computing, quantum computing, or AI in each one of them. To know what we have to fight against, it is at first important to make the invisible enemy visible and to understand the mechanisms of infections of the pathogen. In the case of 2019 nCoV, researchers from the University of Texas at Austin have solved the 3D structure of the glycoprotein, more specifically the spike of the virus, with a technique called cryo-electron microscopy. This spike, also called protein S, is the part which is responsible to bind with the ACE2 receptor of the human host cell, initiating the infection. To prepare the protein to be seen with electron microscopy, we need to freeze it in a thin layer of liquid first. An electron microscope is a microscope that uses a beam of accelerated electrons as a source of illumination. An electron gun shoots electrons at the speed of light, passing through the sample. Then a special high-tech camera captures the electrons to form an image. This is called micrography. This is the experimental part of the process that takes place in the laboratory. In the in silico part of the procedure, it is necessary to process the images taken in the laboratory, which requires heavy computational methods. To get the 3D model of the protein, thousands of molecules need to be generated first. On the slides, you can see the different steps that are necessary in this process. These include particle picking, which is a technique used to obtain individual particles from an image. This can be achieved, for example, through machine learning methods. The next step includes 2D and 3D classification, refinement, sharpening, and finally the modeling and interpretation. The result of the aforementioned step is the 3D model of the macromolecule at a resolution of 3.5 angstrom, which correlates to 0.35 nanometers. As a reminder, this macromolecule is the CoV spike glycoprotein, which is a key target for vaccines, therapeutic antibodies, and diagnostics. This leads us to the following section, which is all about getting ready for the fight. Once researchers have identified a possible target, in this case the spike of the virus, they can test a library of compounds against it. In some cases, the library can be as large as 10 over 60 different drug-like compounds. This process is called drug discovery, and it is the first step towards finding a suitable treatment. The following step is the preclinical testing, when you go to the lab to test the more promising compounds, typically on animals. Then you move to humans for the clinical trials, and finally you have to follow the EMA or FDA approval. Here I'm highlighting 3 main methodologies that researchers use in drug discovery processes. Molecular dynamics simulate the movements of the molecules, which can lead to new discoveries, but more on that on the next slide. Molecular docking simulates the docking of a drug on a potential target. The drug gets tested in different positions to find the best fitting solution. Finally, molecular modeling is a complementary method to the other 2. Here we already know the drug that needs to be used and where it needs to bind. In this step, we try to fit the active ingredients into the right shape. For this, several positions get tested. Of course, all of these simulations are computationally demanding, which can get very expensive very fast. This is the reason why, in the middle of this crisis, several initiatives have arisen to allocate computing resources to crack on the millions of existing compounds against different target molecules. There are many of them, but I'll mention Escalate and Folding at Home. Escalate is a consortium where we as Atos have been asked to participate, while Folding at Home is on us as individuals to contribute with the power of our laptops and computers. In these videos, you can see the results of this at-home folding project. In both cases, the target molecule is a protease, which is key in allowing the replication of the virus. Therefore, many treatment targets at inhibiting this protein. The first video is the molecular dynamic simulation of the protease, revealing a hidden drug binding site. The second video is on docking a drug against that pocket. The final action that will be discussed today is the tracking of the spreading, which is a crucial step when trying to control the outbreak. Whole genome sequencing and analyzing differences in the genetic code of viruses from different patients is used to map the spread of the virus in real time, tracking new mutations to identify if different strains are emerging. Better understanding of the genetic makeup of the virus could ultimately save lives by forming strategies for public health and clinical care, as well as facilitating the design of therapies and vaccines to combat the virus. This already happens, for example, through the Whole Genome Sequence Alliance in the UK, which maps the spread of the coronavirus. It is a consortium formed by NHS, public health agencies, the Sanger Institute, and numerous academic institutions. They distribute the work amongst them, coordinated by the Sanger Institute.
We've come to the end of the presentation. Thank you so much for listening. I really hope you enjoyed it. It is important to note that these techniques are not only relevant in the fight against COVID-19, but similar approaches could also be feasible against other illnesses such as cancer. As the need for expertise in this field is now as high as ever, I hope this presentation also inspired you a bit to become part of the solution. Meanwhile, from my home office to yours, stay safe.